Now we'll move to the next presentation by Dr. Nigil Kantasi. Dr. Dhananjay?
Yeah, thank you, sir. So now we have the presentation by Nikhil Gandasi. So Dr. Nikhil Gandasi is an assistant professor in the Department of Developmental Biology and Genetics, Indian Institute of Science. His research focuses on the cell metabolism and disorders related to cellular functions, contributing to the understanding of metabolic diseases. So thank you, sir, for joining this event. Over to you, sir.
Thank you so much. I hope I'm audible. I'll try to share my screen now.
Yeah, you are audible.
Okay, I hope my screen is visible.
Yes, it's fine. Please go
Okay,
ahead.
good. Great. So I'm a little bit off from the other people, I guess. I do more sort of basic research compared to all of the people that I heard here. So I will try to go very slow and try to make you understand what I'm speaking and why I'm speaking and things like that. I'm going to speak about diabetes. Diabetes has become a lifestyle disorder and it very much impacts aging. So yeah. So if you just look through what is diabetes, how it is contributing and things like that, you will see this kind of a graph where you will see children and adolescents which have very low incidence of diabetes and people who are aged between 20 to 64 having a very high prevalence and the older people also having prevalence. But what is surprising is the 20 to 64 group, which are not really, they're the aging population, right? So they're not really the old population yet, but still the prevalence is quite high there. So one of the things to consider here is that the people don't really go and test themselves. So one of the things I want to highlight here is the HbA1c. So HbA1c is you have the RBCs that you have, and the glucose that is present in the blood will you will deposit more and more here to form a glycated hemoglobin. This is what is measured as HbA1c, hemoglobin which is glycated, and you can see something like this, which is very much glycated. So in a normal individual, so the high HbA1c is below 5.7%. So in a pre-diabetic individual, the HbA1c is above 5.7, can go up to 6.5. And if it's above 6.5, it's definitely diabetic. This is also very different in case of Europe or in case of States or in case of India. So in this context, I'm just speaking about Asian population in this case here. So you also see the blood glucose and which is normal and pre-diabetic and diabetic here. So how often do we see people who are aged like 20 to 40 or 20 to 50 going to hospital and finding out what is their HbA1c levels or finding out how glucose intolerant they've become? So that is one of the reasons why I think diabetes in a younger age is so high. So we need to do something about it. So we are trying to solve this by looking at cells. But before we go into this, I just want to show the prevalence of diabetes and in India especially. So if you look at the prevalence, there are these metropolitan cities that you see like Mumbai, Hyderabad, Chennai, etc., where the prevalence is quite high. More than 15% of the population in each of these cases are diabetic, which is not good at all. And if you just look at the prevalence year on year, around 2000, we had 5.8% of people in India who were diabetic. And by 2015, we were at 8.7%. And today we are more like above 9%. So this is not going very well. And if that is the case, we'll have Indian people having like 134 million people in India itself by 2045 suffering from diabetes. We are an aging population, and it doesn't really help that the number of diabetic individuals increasing means that there is more and more care that has to be taken and more investment that has to be done. Right now, we stand second in terms of prevalence, but where it's China is first, but very soon we'll be first if this is how the prevalence is rising, I think. So I'm telling you a lot about prevalence of diabetes. but I'll just tell you what happens when we have a meal. So today, when you had breakfast, there was an increase in blood glucose. And this increase in blood glucose means that the glucose is being taken up by the pancreas. Pancreas is the organ which is the interest of choice that we study a lot in our lab. So when there's glucose uptake here, there is secretion of insulin that happens. And this secretion of insulin means that the excess glucose that is there in the blood is taken up by all the peripheral tissues like fat tissue, muscle tissue, liver tissue, etc., which is very good because then the blood glucose levels decrease, which is very important to keep this in check so the person is not diabetic. So what happens during diabetes? There are two things that come into play. One is insulin resistance. Insulin resistance means that insulin is being secreted from the pancreas, but then the blood glucose remains high because the peripheral tissues are not taking up glucose. That's one of them. The other is islet cell dysfunction, which means that the pancreas is trying to compensate for the excess glucose that's present in the blood due to insulin resistance, and then they try to overwork themselves, leading to islet cell dysfunction. Most of our focus in the lab has been on islet cell dysfunction. There are a lot many people working on insulin resistance in the world. This is one of the areas that we felt not much is being worked on, so that has been the focus of the lab. So our lab at Indian Institute of Science in Bangalore focuses mostly on pancreas and pancreatic disorders. And pancreas in general is the size of a fist. So it's a very small organ in the body, but it's very crucial because it's important for digestion of what we eat and also to maintain blood glucose levels. So the green dots that you see there in the pancreas, they make up less than 5% of the pancreas, but they are the ones which have the pancreatic islets of Langerhans. So these islet of Langerhans have these green cells, which are insulin secreting beta cells, and also they have other cell types like blue ones, which is glucagon secreting alpha cells, and also pink ones, which are somatostatin secreting delta cells and others. So we focus mostly on green cells here, the insulin secreting beta cells, because insulin is one of the main things in case of diabetes. So this is our lab, which focuses on insulin secreting beta cells in type 1 and type 2 diabetes. So why do this in terms of aging? Why we do this is if you take a section of the pancreas, so this is an younger individual that you see here, and it's an older individual that you see here, and you can see the difference in the tissue here. So there are a large number of atrophic regions in these pancreas that you see. And what is quantified here is that they are really mild in case of like below 60s. And from 70 to 79, they have a large, the changes that you see there, which is from 70 to 79, and this keeps raising. I mean, the data is very less in 90 to 99 and 100 to 109, so just ignore this part. But you can see that the orange bars raise as we age. So that is one of the things that is there in the pancreas. And if you look at aging lens in this perspective, there is something called cellular senescence, where a large number of cells don't really undergo division and they become very silent. And this leads to also very similar things like insulin resistance, metabolic dysfunction, and things like that, which is very similar what is seen in case of diabetes as well, where there is very high glucose. So high glucose can also lead to cellular senescence in aging. That is why aged people have a higher chance of getting diabetes. And that is the reason our lab is focused on looking at metabolic dysfunction, what happens during insulin excitosis, what are the different things that take place during other tissues and complications that are there during diabetes and things like that in an aging perspective. So with this sort of introduction, I will try to go in a little bit deeper into beta cells and tell you what happens. So these beta cells, when there is glucose, they take up glucose and they generate ATP. So this is very important for the normal cell function because there are large number of granules. These are the gray ones that you see there. These are very small. There are like 100 nanometer in the cell, in the beta cells. So these are the ones which have a high quantity of insulin present in them. So these insulin secreting granules have to fuse to the plasma membrane, and this fusion leads to secretion of insulin. And this insulin is then going via the blood vessels to reach the different tissues that I spoke about. You name it, liver, muscle, kidney, all of this requires insulin from the pancreatic beta cells. And these are the granules which are having these insulin inside them. So once the insulin is secreted, then the blood glucose lowering can happen because the blood glucose is being taken up by all the different tissues in the body. And that's what we want from this. So in case of diabetes, what happens is that there is a change in this phenomenon, which I will tell you in the future slides. But how we do this, or how we approach this, or how we study these insulin granules is by using microscopy. So microscopy is one of the very good tools to study single cells, like I said, the single beta cells, because these are the ones which have insulin granules, which are these 100 nanometer structures. They are very small, very hard to visualize. So we employ various different techniques to visualize them. Some of it is called epifluorescence microscope, where you can see the whole body of an islet pancreatic beta cell, or let's say, for example, TIRF microscopy, where we can just focus on plasma membrane of the beta cell. This is important because this is where these granules come, fuse to the plasma membrane of the cell, and release insulin as the content. So this is confocal, where you can take optical sections. Don't worry about it right now. So we focus mostly on the TIRF microscope. If you look at images from TIRF microscope, you will see a fluorescently labeled dots that you see here. These fluorescently labeled dots are the insulin granules present in them. And these insulin granules are the one which undergo exocytosis. So I'm telling you a lot about TIRF microscopy and all these cell types. So what we do is we isolate these pancreatic beta cells, and then we fluorescently label them. So this is how they look. You see a lot of puncta or number of dots there, and these dots are the insulin granules, and we stimulate them with glucose, very similar to when you eat a meal. There is an increase in blood glucose. Exactly same thing also happens here, where we try to challenge them with glucose, where there's very high glucose concentrations, which means that you see exocytosis. So this exocytosis looks something like this, where a large number of granules come to the plasma membrane, and you see some sort of a fusion event there. So you can see an increase in intensity and a large number of granules disappearing from the field. This disappearance is because they're coming to the plasma membrane, they're fusing to the plasma membrane, and then they're releasing insulin into the blood vessels of the cell. So this means that there is single individual events which look like this, which are the granules present in the plasma membrane, and then they're releasing, which means the disappearance in the TIRF field. So, okay. So this disappearance means that whatever granules are lost here during exocytosis, all of them have to be replenished. So there should be more granules coming to the membrane, and they should dock there. So this is what we call as docking, where the cytosolic individual insulin granules come to the plasma membrane of the cell, and they undergo a process called docking, which is where they get stabilized. So we should visualize this as well, the docking, where the granules are not present in the TIRF field. They are completely absent. And then you see individual granules coming and sticking there, right? So when we had access to a large number of cadaveric human islets, we tried to image them. So these are human islets that you see there. These dots are the insulin granules, and you see some circles there, which are the number of docked granules. So the number of granules which are there at the plasma membrane of the cell. And like I showed you in the previous slide, we challenge them with glucose, which means that there is insulin exocytosis, means insulin is being secreted out to the blood vessels. So this is what you see by the end of the experiment. So this means that you can measure the total number of exocytotic events then. So from each individual, we take like 20 cells to see this. And then you see the dot that is there, which is each individual that you're seeing there. This each individual, we plot the density of docked granules, the density of number of granules that you see there, and the total number of exocytotic events that you see there. So in this case, you see type 2s, which are in the lower version here. So these are all diabetic people. So they are all plotted with a lower density of docked granules, which means that there is less exocytosis. So we went ahead and we started seeing individual sort of cells in the individuals. So you see each dot here, again, a single individual. So each dot is an individual. So you see a lower density of these dots or dot granules that you see in case of type 2 diabetic. In case of non-diabetic, you see a higher density of dot granules. So you can see on an average as well, this is much higher compared to type 2 diabetes, which is much lower. So this was one of the things where we saw that the total number of dot granules was reduced in case of type 2 diabetes. This is very crucial because when this happens, this can correlate very well with the insulin secretion that you see in the peripheral blood. So this is insulin biphasic response that we see in the peripheral blood. You see the fast first phase and the slow second phase. This is there in a non-diabetic individual. In case of type 2 diabetic individual, you don't really see the biphasic response. So this is in the peripheral blood, and we look at individual cells, there as well we can see a sort of biphasic secretion. So this is important because the total number of insulin granules, they deplete over time, which is not at all available in case of type 2 diabetes, like I showed you in the previous slide. So the biphasic response that I showed you is all because of glucose. So we also work on how glucose is being taken up by the pancreatic beta cells so that the insulin secretion can be initiated. So one of the students in the lab is working on glucose transporters. In what she sees is that in case of type 2 diabetic individuals, the total number of glucose transporters at the plasma membrane, they are also very few. Why is this important? This is important because the GLUT transporters have to be recruited to the membrane for them to take up glucose. If they can't take up glucose, then it's a problem, which is the case in case of type 2 diabetes, which also means there's a reduced in expression. So there are two things from what I've been talking about. One is the non-diabetic individuals where they have glucose transporters, where glucose is taken up, and then there is glucose leading to insulin secretion, which is released like this. And if there is less number of transporters in case of type 2 diabetic individuals, less of glucose is being taken up, which means that there is less of insulin exocytosis that take place. So we tried to see if we can use this for therapeutic purposes. So one of the postdocs in the labs at the Deepa has been working on this. So she found out that a compound called FU4 A can increase the amount of time these GLUT transporters spend at the plasma membrane of the cell. This is very important because if you arrest these transporters, that means that more and more glucose can come into the cell, which means it helps in the secretion of insulin. So that's all about insulin secretion and how this biphasic insulin secretion is triggered by glucose. So I'm telling you a lot about triggered by glucose. There's one more aspect to this. When there's an increase in glucose, there are calcium channels which take up calcium, and this helps in a case of a release of insulin. And in case of type 2 diabetic individuals, as you can see, there is reduced number of insulin secretion. There's also reduced degree of calcium channels. So you see that there is multiple levels that you see: glucose transporters, calcium channels, or let's say insulin granules. All of these are dysregulated in case of type 2 diabetes. So we wanted to find reasons for this. So we looked at the type 2 diabetic individuals to see some of the proteins that are important for docking, that is important for calcium channel recruitment. And some of them are REM, RAB, etc. Their expression at the plasma membrane is also reduced in case of type 2 diabetes. So here we are trying to find molecular cues for, or the reasons for what is happening. Why do we have less granules? Why do we have less calcium channels? Or why do we have less glucose transporters? So these could be some of the molecular cues. So one of them was Syntaxin in this case. And the Syntaxin, what it does was important for us. We looked at super resolution microscopy, where we can see individual molecules of Syntaxin that are labeled here in green, and the red ones are the insulin granules. What you can appreciate is that the Syntaxin forms a cluster there, which helps in recruitment of insulin granules. That is exactly what is plotted here. I'm not going into the details of it in the interest of time. So there are many, many proteins that we looked at as molecular cues, and what these molecular cues do for the insulin secretion to happen. We have found various proteins apart from Syntaxin and calcium channel that I was describing about, Syntaxin 3, RAB7, complexin, alpha SNAP, etc. All these, we found what it does and what is different in case of type 2 diabetes. So what we tried to do is, since we know some of these proteins, there is less expression, we took the pancreatic beta cells from the type 2 diabetic individuals and increased the expression of these proteins. So when you increase the expression of these proteins, just as an example, in this case, Syntaxin, you also see an increase in the density of dot granules, which also means that there is increased recruitment of calcium channels. I told you 3 problems. One is dot granules, other is calcium channels, other is GLUT transporters. So with Syntaxin 1, we could solve 2 of the problems, which is very crucial because we know a way to do this. And we tried this with various other proteins where we also saw an increase. This is important because in a clinical setting where you have like 200 individuals and you try to see HbA1c, meaning how diabetic an individual is, and correlate this with the expression of these proteins, there is a very marked inverse correlation, which means the higher HbA1c means there is less of expression of these proteins. So that also means there is less of insulin secretion that's happening in these individuals. All in all, we have sort of cues of what these proteins could do and at a cellular level and what is leading them to form diabetes. So we want to know how we can increase it by giving sort of drugs and things like that. So one of the drugs that we were interested in was EPAC2. The EPAC2 is not a drug, EPAC2 is a protein. GLP-1 and GLP-1 analogs are drugs. So if you're diabetic, if you're aged, and if you're going to a clinician, they normally prescribe you GLP-1 analogs apart from metformin. So here what happens is EPAC2 gets activated, which is normally the case in case of non-diabetic individuals as well, which leads to more insulin secretion. In type 2 diabetic individuals as well, you can see an increase in insulin secretion, but if you correlate this with HbA1c, which shows how diabetic a person is, there is only to a certain extent that this can increase the secretion. But if the person is severely diabetic, which you see here, over 6.6 or something like that, this drug doesn't do anything. So this is also important to know. Let's say an individual, how diabetic an individual is, how aged an individual is, and what kinds of drugs that you prescribe, and what happens when you look at single cells and what are the changes they bring out. This is what we can find out from this. So in summary, we have been looking at individual pancreatic beta cells to see what they do and how they secrete insulin and how they bring those insulin granules to the plasma membrane and how they involve calcium in the process to lead to insulin exocytosis. And in case of type 2 diabetes, both the docking, which I described, and the priming, which is recruitment of calcium channels, both of this is stopped, which means there is less of insulin exocytosis that is taking place, apart from, of course, GLUT transporters as well, which is decreased. So, but we have found some of the targets which can initiate more and more calcium channel recruitment, which one of which I was describing, which is GLP-1 analogs. But there is more and more to do. That is where I think there is potential. So with this, I would say that I've focused most of my talk on insulin secretion and beta cells, but there is also alpha cells in this context, which is very important for hyperglycemia, and also delta cells, which is also important for control of alpha and delta cells. I think I will not spend much time because I realize I've spent my time trying to discuss about beta cells. So I'll skip many of the things that I was going to say in future, and I'll just end by saying that it's important to look at beta cells and to understand what is happening in case of beta cells and how we can avoid getting dysfunctional beta cells as we age. And glucagon secretion is for another day to discuss, and there's a lot more to do here, and also some extra secretion. So this is what our lab does, and these are the funding agencies which fund us, and all the people in the group who have contributed extensively to do this, and all the donors where we have been getting pancreatic islets, and Longevity India, which has also been a great support. Thank you so much.
Thank you, Dr. Nigel, for that highlighting some of the very important research you and your team doing in your lab, particularly on diabetes and its implications for the overall health and well-being of the people.
Thank
We
you.
love